Assalamu alaikum and welcome again to the medical concept with Dr. Falak. In today's video, we are going to discuss red blood cells. They are also known as erythrocyte. They are the most abundant cell in our blood, specially designed for oxygen transport. So we will see how their unique structure, membrane and internal organization allow them efficiently carry oxygen and survive in circulation. By the end of this lecture, you all should be able to describe the red blood cell structure and its membrane component. You should be able to explain the function of key membrane proteins, differentiate vertical versus horizontal interaction, outline red blood cells life cycle and metabolism, interpret red blood cells indices and classify anemia, explain the function of red blood cells, understand the regulation of erythropoiesis. Red blood cells are the flexible, biconcave, anucleated cells that contain hemoglobin, and their main function is primarily to deliver oxygen and carbon dioxide. In this slide, we are looking at the composition of a red blood cell, and one key idea you should keep in mind is that red blood cells are highly specialized by losing many of their organelles. First, notice what is absent. Red blood cells do not have a nucleus, ribosome, endoplasmic reticulum or Golgi apparatus, so they cannot synthesize protein or repair themselves. They also lack mitochondria, which means no aerobic metabolism, and all of their energy comes from the anaerobic glycolysis. In addition, lysosomes and peroxisomes are absent, so no intracellular digestion and typical detoxification pathways are present. So instead, red blood cells rely on the HMP shunt to generate NADPH for protection against oxidative damage. Now coming to what is present. The cytoplasm is mainly composed of about 60 to 65% water and roughly one-third hemoglobin, which is essential for oxygen transport. Along with this, there are a small amount of electrolytes and important enzymes, including glycolytic enzymes, carbonic anhydrase, and other antioxidant systems like glucose 6-phosphate dehydrogenase pathway enzymes. So overall, the key concept is that red blood cells has removed almost everything non-essential to maximize space for hemoglobin and optimize its function in gas transport. In this slide, we understand why hemoglobin is enclosed inside red blood cells instead of freely circulating in the plasma. In some invertebrate, hemoglobin exists as a free protein in circulation, but in human, this would be highly inefficient and even harmful. Encapsulation of hemoglobin inside red blood cells serves two main important purposes, right? First, it prevents loss of hemoglobin. If hemoglobin were free in the plasma, a significant amount would leak out during circulation, both through capillary walls into the tissues and through glomerular membrane into the urine. Second, it ensures efficient oxygen transport. By packaging hemoglobin inside red cells, the body can carry a much higher concentration of oxygen without increasing plasma viscosity or losing proteins. So the key takeaway is that enclosing hemoglobin within red blood cells is essential to prevent its loss and to optimize oxygen delivery. In this slide, we are looking at the shape and the size of the red blood cells. Red blood cells have biconcave disc, which is very important structural feature. This shape increases the surface area for gas exchange and also allows the cell to deform easily while passing through narrow capillaries. In terms of size, the average diameter of a red blood cell is about 7.8 micrometer. The thickness is not uniform. It's around 2.5 micrometer at the periphery and less than 1 micrometer at the center, which gives the cell its characteristic biconcave appearance. The average volume of a red blood cell is about 90 to 95 cubic micrometer, which corresponds to mean corpuscular volume or MCV. So overall, both the shape and dimension of the red blood cells are precisely optimized to maximize oxygen transport and ensure smooth flow through microcirculation. Deformability refers to the ability of the red blood cells to undergo reversible shape change so they can pass through capillaries that are even smaller than their own diameter. Functionally, red blood cells behave like a flexible bag-like structure rather than rigid cells. This property is possible because red blood cells have excess membrane relative to their internal volume, which allow them to undergo significant deformation without rupturing. As a result, red blood cells can easily squeeze through very narrow capillaries, sometimes as small as 3 to 4 micrometer, without getting damaged. This prevents mechanical energy during circulation and ensures efficient delivery of oxygen to the tissues. So deformability is essential for both red blood cell survival and the proper microcirculatory flow. This deformability and the shape of the red blood cells are maintained by a specialized membrane skeleton that is present just beneath the lipid bilayer. The red blood cell membrane has two main components. First, the lipid bilayer with transmembrane proteins such as band-3, glycophorins, Rh factor, aquaporin, and GLUT1 transporter. These proteins are mainly involved in the transport and surface function. Second, the underlying cytoskeletal network, which include spectrin, actin, ankyrin, protein 4.1, and protein 4.2. This network provides mechanical strength and flexibility to the cell membrane. The interaction between these two components, membrane proteins and the cytoskeleton, is what allows the red blood cells to maintain their shape while remaining highly deformable. Now focusing on the major transmembrane protein of the red blood cell membrane. First, band-3, also known as the anion exchanger. Its main function is to exchange chloride and bicarbonate ion, which is essential for carbon dioxide transport in the blood, that is the chloride shift. In addition, band-3 plays a structural role by anchoring the cytoskeleton to the membrane through ankyrin. Next, GLUT1. GLUT1 facilitates passive bidirectional transport of glucose. Since red blood cells depend entirely on the glycolysis for energy, this transporter is absolutely essential for their survival. Aquaporin 1, which allows rapid movement of water across the membrane. This helps maintain osmotic balance and allow red blood cells to adapt quickly to the changes in the environment. Now moving to glycophorin, which are another important type of transmembrane proteins of the red blood cell membrane. Glycophorin A is the most abundant. It is rich in sialic acid, which gives the red blood cell a surface, a negative charge. This negative charge is very important because it prevents red blood cells from sticking together and maintain smooth flow in circulation. It also carries important blood groups antigen. Glycophorin B. It's structurally similar to the glycophorin A but is less abundant. Its main clinical importance is that it carries the SS blood group antigen with only a minor role in surface charge. Glycophorin C has different role. It is more involved in structural stability. It connects with protein 4.1 and helps stabilizes the interaction between the membrane and the cytoskeleton, contributing to mechanical integrity of the red blood cell membrane. Glycophorin D is essentially a shorter form of glycophorin C and shares similar function, though its role is relatively minor. So overall, glycophorin A and B are more important as surface properties and blood grouping, while glycophorin C and D are more important for structural support. Now looking at the cytoskeletal network of the RBC membrane, which is responsible for the strength and flexibility, spectrin is the main structural protein. It forms a long, flexible mesh-like network beneath the membrane as alpha and beta chains, giving the red blood cell its elasticity. Actin forms a junctional complex and it connects spectrin filament, helping maintain the network structure. Ankyrin acts as a linker protein, anchoring spectrin to membrane proteins like band-3, thereby connecting the cytoskeleton network to the lipid bilayer. Protein 4.1 plays a key role in stabilizing the spectrin-acting junctional complex. It strengthens the horizontal interaction with the cytoskeleton and helps maintain the shape and flexibility of the cell. Its defects leads to hereditary eleptocytosis. Protein 4.2 on the other hand is involved in the vertical interaction. It association with the band-3 and stabilizes the ankyrin band-3 linkage, helping maintain membrane integrity and surface area. Its defect is associated with the hereditary spherocytosis. So overall, the coordinated action of these proteins allow red blood cells to remain both stable and highly deformable. Now look at this image. This image is showing you the membrane bilipid layer of a red blood cell. Then comes the certain transmembrane protein that we have just discussed. It includes Rh factor, glycophorins A, B, C and D, band-3 protein, GLUT transporters and aquaporins. Apart from these transmembrane proteins, we have also certain cytoskeletal components that include ankyrin, protein 4.1 and 4.2, and actin along with the major spectrin alpha chain and spectrin beta chain. note that transmembrane proteins like Rh factor, glue transporter, aquaporin, they occupy a specific position in their membrane bilipid. This is also true for glycophorin A, B, D, and C. Then comes the band 3 protein, as we have discussed previously. This band 3 is connected with the spectrin chain by means of N-chirin, a cytoskeletal protein. This connection is further supported by means of protein 4.2, which stabilizes this bond. Then comes the actin filament. This actin filament is responsible for connecting glycophorin C with the spectrin molecules. The spectrin molecule and the actin filament connection or the junction is further strengthened by means of protein 4.1. This is actually the point that is known as junctional point, and here you will find that 4 or 5 structural chains of alpha and beta spectrins are connected, and thus they provide a cytoskeletal network throughout the red blood cell membrane. Summary: red blood cells, the membrane stability depend upon two types of structural interactions, the vertical interaction and the horizontal interaction. Vertical interactions connect the cytoskeleton to the lipid bilayer through proteins such as spectrin, N-chirin, band 3 and protein 4.2. These interactions are responsible for maintaining membrane attachment and surface area. When vertical interactions are defective, part of the membrane are lost, leading to a decrease in surface area and the formation of spherical red blood cells as seen in hereditary spherocytosis. In contrast, the horizontal interactions occur within the cytoskeleton itself, mainly between the spectrin, the actin, and the protein 4.1. These interactions maintain the internal network and provide flexibility and shape stability. When horizontal interactions are defective, the cytoskeletal network becomes weak, causing the red blood cells to elongate into elliptical shape, resulting in hereditary elliptocytosis. Now let's understand the lifespan of red blood cells. Red blood cells are produced in the bone marrow and they are released into the blood as an immature radiculocytes. Later on they mature into fully functional red blood cells. Their average lifespan is about 120 days. During circulation, red blood cells continuously pass through capillaries and the spleen. Over time, they gradually loses enzymes and their ATP, which leads to reduced membrane flexibility and decreased deformability. As RBC ages, they become more rigid and less able to pass through the narrow splenic sinusoids. These senescent red cells are then removed by macrophages mainly in the spleen but also in the liver and the bone marrow. This process is known as extravascular hemolysis. After destruction, hemoglobin is broken down, globin is reused as amino acid, iron is recycled and heme is converted into biliverdin then bilirubin. So the entire life cycle from production to the destruction is a continuous, well-regulated process essential for maintaining healthy red cells function. Now coming to the metabolism of red blood cells. Red blood cells lack mitochondria, so they cannot perform aerobic metabolism. As a result, they rely entirely on the anaerobic glycolysis for their energy needs. The main path is the Embden-Meyerhof pathway, which generates ATP. This ATP is essential for maintaining membrane integrity, ion balance, and overall cell survival. Another important pathway is the hexose monophosphate shunt, which produces NADPH. NADPH plays a critical role in protecting RBCs from oxidative damage by maintaining glutathione in its reduced form. Clinically, this becomes important in glucose 6-phosphate deficiency. In such cases, RBCs cannot generate adequate NADPH, making them more susceptible to oxidative stress and hemolysis. So red blood cells metabolism is simple but highly specialized, focused on energy production and protection against oxidative damage. Now coming to the concentration of red blood cells. The average number of red blood cells per cubic millimeter varies slightly between males and females. In healthy men, it is about 5.2 million, while in healthy women, it is around 4.7 million with slight variation. Another important point is that people living at higher altitudes tend to have a higher RBC count. This is a physiological adaptation to lower oxygen level, where the body increases red blood cell production to improve oxygen carrying capacity. So red blood cell concentration is not fixed. It varies with factors like sex and environmental conditions such as altitude. Now focusing on hemoglobin concentration within red blood cells. Red blood cells can concentrate hemoglobin up to 34 grams per 100 milliliter of cells. This represents the maximum metabolic limit of hemoglobin synthesis, meaning the cell cannot pack in more hemoglobin beyond this level. Under normal condition, red blood cells are almost fully saturated with hemoglobin, which allows optimal oxygen carrying capacity. When hemoglobin is deficient, the concentration of hemoglobin inside RBCs decreases. As a result, the cell becomes smaller, and this is called microcytosis because there is less hemoglobin available to fill the cell. So hemoglobin concentration not only determine oxygen transport but also directly influence the size of the red blood cells. Now coming to the hemoglobin content in whole blood. Hemoglobin concentration is closely related to the hematocrit, which is the percentage of the blood volume occupied by red blood cells. Normally, hematocrit is around 40 to 45%. In terms of hemoglobin level, healthy men have about 15 grams per 100 milliliter of the blood, while women it is slightly lower, that is around 14 grams per 100 milliliter. Another important concept is the size of RBCs in relation to the hemoglobin content. Based on the cell size, RBCs can be classified as normocytic, microcytic, or macrocytic. Microcytic cells are smaller and are usually associated with decreased hemoglobin, while microcytic cells are larger and often seen in certain deficiencies like vitamin B12 or folate. So hemoglobin content not only reflects oxygen carrying capacity but also helps in classifying different types of anemia. Now coming to the functions of the red blood cell. The primary function of red blood cell is to carry hemoglobin. Hemoglobin contains heme, which has iron, and this allows it to bind oxygen. This hemoglobin then carries oxygen from lungs to the tissues where it is released for cellular use. So essentially red blood cells act as a carrier and hemoglobin is the molecule that actually performs oxygen transport. Now looking at the additional function of red blood cell, which is carbon dioxide transport. Red blood cell contain the enzyme carbonic anhydrase, which plays a key role in this process. It catalyzes the reaction between carbon dioxide and water to form carbonic acid, which is then dissociate into hydrogen ion and bicarbonate. The hydrogen ion binds to hemoglobin while bicarbonate moves out of the cell in exchange for chloride ion. This is known as chloride shift. An important point is that carbonic anhydrase increases the speed of this reaction thousands of times, making carbon dioxide transport efficient enough to meet the body's need. So in addition to the oxygen transport, RBCs also play a major role in carbon dioxide transport and acid-base balance. Continuing with the role of RBCs in carbon dioxide transport, most of the carbon dioxide in the blood is transported in the form of bicarbonate. Inside red blood cells, carbon dioxide from the tissue is converted into bicarbonate, which then moves out in the plasma in exchange of chloride ion. This allows blood to efficiently carry carbon dioxide from tissues to the lung. When the blood reaches the lung, the process reverses. Bicarbonate re-enter the red blood cells, combine with the hydrogen ion to form carbonic acid, which is then converted back into carbon dioxide and water. The carbon dioxide then diffuses into the alveoli and is expired out. So RBC play a central role in transporting carbon dioxide in a reversible and highly efficient manner. Another important function of red blood cell is their role in acid-base balance. RBC acts as a buffer, helps to maintain the pH of the blood within a narrow range. This is mainly due to hemoglobin. Hemoglobin is a protein with amino acid side chain that can either bind hydrogen ion or release them. In this way, it can act both as an acid and a base, which make it as amphoteric buffer. By binding excess hydrogen ion or releasing them when needed, hemoglobin helps stabilize blood pH despite ongoing metabolic activities. So in addition to the gas transport, RBCs plays a critical role in maintaining acid-base homeostasis. Now let's understand the regulation of red blood cell production. Red blood cells are produced in the bone marrow from hematopoietic stem cells. The most important stimulus for their production is hypoxia or low oxygen level in the body. In response to hypoxia, the kidney, along with the liver to a lesser extent, secrete erythropoietin. This hormone stimulates the bone marrow to increase red blood cell production. In addition, various cytokines such as interleukin, including interleukin 1, 3, and 6, also support erythropoiesis. Another important factor is granulocyte monocyte colony stimulating factor, which helps in the development and proliferation of erythroid stem cells. So red blood cell production is tightly regulated process, mainly driven by oxygen demand and mediated through erythropoietin and supportive growth factor. Now coming to the red blood cells indices, which are the quantitative parameters used to describe the size and hemoglobin content of the red blood cells. These are routinely calculated in blood tests and are very important in evaluating anemia. Mean cell volume or MCV represents the average volume of a red blood cell. It helps classify red blood cells as microcytic, normocytic, or macrocytic. Then we have mean cell hemoglobin or MCH. It indicates the average amount of hemoglobin present in each red cell. Then mean cell hemoglobin concentration or MCHC, that reflect the concentration of hemoglobin with a given volume of packed red blood cells. Finally, red cell distribution width, that shows the variation in the size of red blood cells, and increased indicate greater variation, which is helpful in differentiating different types of anemia. So red blood cells indices provide a structured way to analyze red cell morphology and are essential for clinical interpretation. Now a quick rapid review of the red blood cell morphology abnormalities. Anisocytosis means variation in the size, while poikilocytosis means variation in shape. Microcytosis refers to the smaller cells and macrocytosis to the larger cells. Hypochromia indicate reduced hemoglobin, so the cells appear pale. Spherocytes are spherical cells without central pallor, while elliptocytes are elongated. Target cells show a bullseye appearance. Schistocytes are fragmented cells and acanthocytes have irregular spiky projections, whereas echinocytes have uniform projections. So remember, size change, shape change and hemoglobin content together help you quickly identify different red blood cells abnormalities. Now let's do an important clinical task, the interpretation of complete blood count report. Here in this picture you will find that hemoglobin is 8.2 gram per deciliter, which is clearly low and thus indicating anemia. The hematocrit is also reduced at 28%, that also support this finding. Looking at RBC indices, the MCV is 64 femtoliter, which indicate microcytosis. MCH is also low, suggesting reduced hemoglobin content per cell. MCHC is relatively within range, but overall the picture points toward hypochromic microcytic anemia. The total RBC count is within the lower normal range, which is important because in some conditions like thalassemia, RBC count is often relatively higher despite low hemoglobin, but here it is not. The peripheral blood smear further supports the diagnosis, showing anisocytosis, hypochromia, microcytosis, target cells, and elliptocytosis. Putting all these findings together, this is more consistent with iron deficiency anemia, and as suggested in the report, the next step would be to confirm with an iron profile. So this is the classical example of how RBC indices and peripheral smear together help in diagnosing the type of anemia, and the anemia we will be discussed in the next upcoming video. So to conclude, red blood cells are highly specialized cells that are designed to maximize oxygen delivery while also playing important role in carbon dioxide transport and acid-base balance. Their unique structure, membrane protein and metabolism all work together to maintain the flexibility, stability and function throughout their lifespan. Understanding these concepts not only helps in building a strong physiological foundation but also allow us to interpret clinical conditions like anemia more effectively. Thank you for watching. Allah Hafiz.